I'm going to run through some things very quickly. As mentioned, okay, I'm already stuck. There we go. Okay. As mentioned, I am president of the Society of Critical Care Medicine. That's one of those be careful what you wish for jobs. You are forced to listen to me go through disclosures, so here they go. We have a book. You all ought to buy it. It's a really good book. Patents pending on some of the material we'll discuss, but no money. Stockholding a bunch of stuff that isn't doing so well. So I have no real conflicts, but here's the big major problem is these three in grad school and private universities. So I have no conflicts, but I also have no money. So as an SCCM officer, I'm going to stay that way for a while. Now, I want to start by posing the problem in this way. This is very recent stuff from Jack Awashna, a friend of mine at Michigan, looking at the incidence and costs of care in critical illness and in sepsis in particular. And what you can see from this graph is that the incidence of severe sepsis is going up. The cost is going up dramatically. These are data from the U.S. Medicare database, so patients over the age of 65. As opposed to, for example, to acute myocardial infarction, where the incidence is about flat. The cost is actually going down. There's something at the bottom that says people should be appraised of these data before they decide what specialty to go into. Maybe less of them would go into cardiology. The question I want to start by asking is, why is it that nothing we have tried to treat sepsis and multiple organ dysfunction syndrome has worked? And what I will ask you to do, back when I was a kid, there was a TV show called The Outer Limits. And what The Outer Limits did at the beginning of the show was ask you to suspend your beliefs for 30 minutes. So I'm going to ask you to do the same thing. It'll be less than 30 minutes because we don't have commercials. I would point out also here in the corner, this is William Shatner, Captain Kirk, before he was Captain Kirk. So suspend your beliefs for 30 minutes and let's look at things. How do we really describe the pathogenesis of sepsis? Well, there's some kind of injury. You get an acute stress response. It's followed by a systemic inflammatory response syndrome and perhaps a compensatory anti-inflammatory response syndrome, septic shock or severe sepsis, inflammation, immunosuppression, MODS or maybe no MODS, and if there's no MODS, maybe patients get better or die of septic shock. More often, though, what happens is that they end up like this. But that said, suppose you'd never heard of sepsis, and you went to work in an ICU with a lot of septic patients and you watched them for a year. Is what you would see the same as what I just described? And I would suggest, based on, in part, what Dr. Marshall has just presented, that the answer is no. What you would see is something that looks sort of like this. But you'd have patients who die from septic shock, much less than they did many years ago, but they still die from septic shock. But more often what you'd see is some who develop organ failure and then go on to develop something that, for lack of a better term, we'll call chronic critical illness. They are in the ICU, they're on all the machines and interventions that Dr. Marshall showed you pictures of, and some of them get better and some of them die. This is a little different than the way we have always described the syndrome, and I want to look at this a little more closely. So, it really represents four distinct processes. The first one is SIRS or CARS, leading to some abnormality in the inflammatory system. The second is these abnormalities, most often inflammation, leading to septic shock or severe sepsis. The third is septic shock leading to MODS. And the fourth is MODS leading to chronic critical illness. So, what have we studied? Well, we spent a great deal of time and a great deal of money studying this part of the response. But when you really stop and think about it, most of our patients die when they end up in an intensive care unit on all kinds of interventions and somebody decides that enough is enough and we turn off the machines. And yet this is the part that we haven't looked at very carefully. So what I'm going to ask today is, what happens if we look at this particular aspect of things a little more carefully? We've always assumed that septic shock, severe sepsis, MODS, chronic critical illness are pathological responses to exuberant inflammation. Why do we assume that? Well, two sort of things that are concentrated on uncontrolled inflammation and a pathologic response to inflammation. For this talk, I'll talk about the inflammation. The next talk, we'll talk about the pathologic response and whether it's really pathological. Why do we think that these entities result from uncontrolled inflammation? Well, the main reason is about the same time that we discovered the syndrome that Art Bowie called MOF, some other people called MOSF, that we now call MODS, we discovered cytokines. TNF, with septic shock, interleukin-1 with shock-like states, and of course, because these two things were going on at the same time, they had to be involved with each other. That is, the cytokines had to be causing the sepsis, the organ failure, etc., etc. So, here again we have the paradigm. Because the cytokines were recognized at the same time that we recognized MODS, we assumed that they were cause and effect, and we have had all kinds of therapy to reduce mortality that targeted inflammation, assuming this relationship. But interventions that have been directed at altering inflammation, and there have been many, steroids, ibuprofen, anti-endotoxin antibodies, anti-cytokines antibodies, have not worked, including Xigris, which is now gone. And in fact, the most recent studies, a little bit scary, a TLR4 antagonist for adults has not worked. Lactoferrin for neonates, large randomized controlled trials, no difference in outcomes. This is more than a little bit scary because lactoferrin is mother's milk, and if that doesn't work, we are in big trouble. So, to quote one of my heroes, Albert Einstein, insanity is repeating an experiment again and again and again and expecting a different result. And that's what we've done in treating inflammation. So let's for the moment put aside the issue of excessive inflammation and think about what else could be going on. And again, we come back to this paradigm. What else could be going on here? Let's look at the transition from MODS, from organ dysfunction, to chronic critical illness. And think a little bit about what happens here. And in fact, one of the best descriptions was a paper written quite a number of years ago by my friend and nursery school classmate, Tim Buckman, looking at biological oscillators as a pathophysiologic event in sepsis. What Paul Godin and Tim Buckman postulated is the following. Under normal circumstances, everything talks to everything else. The lungs know what the heart is doing, the kidneys know what the liver is doing. They're all connected, they all work together. When you get to MODS, nothing talks to anything else. They're all acting independently and separately. They're not integrated at all. We've lost the integration. We have biological oscillators that are uncoupled. This uncoupling is the characteristic, defining feature of this chronic critical illness. It's the reason that we have to support, in part, all these different organ systems. Under normal circumstances, how does this dialogue work? How are all these different organ systems connected to each other? Well, there are three systems that connect them. The first is the white blood cell system. The second is our hormone, our endocrine system, and the third is the central nervous system. As we all know, the white cell response in this particular state has been looked at quite intensively by a number of different investigators. These are data from Richard Hotchkiss's lab, basically looking at different types of white cells in patients who died of either sepsis or MODS or from trauma, non-septic injury. And these are the patients who died of non-septic injury, staining for inflammatory cells. What you can see is that there's lots of staining here. Now, I'm not an immunologist and I wouldn't know what they're looking at. But over here you can see there's a lot less staining. And we can go back and forth lots of times, and even a non-immunologist can tell that there's a difference between these two. And in fact, if you look at this a little more carefully, as Richard Hotchkiss has done, what you find is that the overwhelming characteristic of inflammation is white cells that are either not present or simply don't work. Energy. The white cell response is lost. The biological oscillators are uncoupled. What about endocrine responses? Well, these have been looked at as well, too, primarily, actually, by Gret Vandenberg's group in Belgium. She is, of course, best known for intensive insulin therapy, but when you look at some of the work that she's done with endocrine responses and her group have done, it really very well defines some of the characteristic changes. So under normal circumstances, you have some signal coming out of the central nervous system via the hypothalamus and perhaps the pituitary gland. It affects something peripherally, that releases something else and goes to a target tissue, and the target tissue responds. So, for example, if the response is coming out of sympathetic ganglia, you get vascular smooth muscle contracting, you get cardiac responses. If it's coming out of immune tissue, you get immune cells that are stimulated. If it's endocrine tissue, you have effects on liver and muscle, etc., etc., etc. And this combination of the peripheral effector and the tissue response we call the peripheral response. Early endocrine dysfunction in our critically ill patients looks exactly like this. When you have a very decreased tissue response, the natural response of both the peripheral effector and the central nervous system will be increased. That is, you're not getting the response you want from the tissue, so you put out more of the substance that's supposed to get the response from the tissue. There's a loss of diurnal variability and this decrease in response. And this is characteristic of stress, early sepsis, or early SIRS. But a little later on, something different happens, which is to say the outflow from the central nervous system, from the hypothalamus and the pituitary, decreases remarkably. And this ends up with a decrease in the stimulation of the peripheral effector and a markedly, even more decreased tissue response. This we would say is MODS, or chronic critical illness, and its central suppression. So let's look at some data. This is TSH, thyroid stimulating hormone, coming out of the pituitary. And what you can see in this work from Vandenberg's group is that under normal circumstances, in the first panel, there is lots of variability and relatively high levels of TSH. That in the acute phase, you lose the variability, although the levels remain somewhat closer to baseline. But in the chronic phase, you end up with very low levels of TSH coming out and almost no variability at all. Again, classic for the initial peripheral response and the central response that comes a little bit later. The same thing happens when you look at growth hormone. Again, this is the normal response, diurnal variability, levels that go up and down and up and down. In the acute phases of critical illness, the variance goes down and the overall levels go down as well, and later the levels go to almost zero. Looking at thyroid dysfunction is a very good example of this. Again, this is a cartoon from Vandenberg's work. If we look at the early response, normally you would have thyroid releasing hormone stimulating TSH, TSH stimulating the release of T4 from the thyroid gland, and then T4 being converted to the active form, T3, in the peripheral tissue. What happens initially in the early phases is that T3 levels are found to be markedly decreased, primarily as a result of a failure to convert T4 to T3 in peripheral tissues. The net response, of course, is that there's an increase in the stimulation to produce more T4. And this is the acute phase, changes in peripheral metabolism and binding. Later on, in the chronic phase, you see decreased hypothalamic pituitary activity, decreases in TRH, TSH, and T4, and a markedly decreased level and activity of T3. This is characteristic of the chronic phase with a neuroendocrine component. Go through this quickly. Similar things for the growth hormone IGF-IGF-BP dysfunction, binding protein. Acute phase, you see decreases in the release of IGF from the liver and in the activity of growth hormone on skeletal muscle and adipose tissue. The net effect, of course, is increased release of growth hormone. Later on, hypothalamic pituitary depression and a markedly decreased response in the periphery. Similar responses have been described for corticosteroids, for sex steroids, catecholamines, glucagon, prolactin, and insulin. So, MODS could be an endocrinopathy. Different than what we've always suspected, not necessarily an inflammatory disease, something completely different. How about altered CNS responses? The only thing that's really in the literature in this regard is the work by Kevin Tracy and his co-workers looking at the vagus nerve. And what they've discovered is that there are stimuli coming up the vagus nerve, afferent flow into the dorsal motor nucleus, that eventually reaches a cholinergic brain network. The net response is an increase in cholinergic outflow down the efferent vagus nerve, decreased inhibition of cytokine release, via an ACTH receptor. The net effect is a balanced response. That is, the initial inflammatory response gets balanced by the outflow from this anti-inflammatory vagal reflex. But what happens when things get unbalanced? Well, when things get unbalanced, when something goes wrong, you don't get the stimulus going up the vagus nerve. You don't get the dorsal motor nucleus and brain stimulus. You don't get vagal currents going downward. You don't end up with the release of the acetylcholine and the inhibition of cytokine release. And the net effect, of course, is markedly increased cytokine release and more tissue injury. This is the CNS as part of the inflammatory response. So on the one hand, we could say that this is brain function or brain dysfunction, but it seems to be working through modulation of the inflammatory response. And again, looking at it a little bit more closely, what you can see here is looking at things up close, that within the spleen, where the vagus terminates, you get stimulation by acetylcholine, of specific acetylcholine receptors. The net effect is inhibition of NF-kappa B activation and decreased cytokine release. Absent that, something will not work properly. You get uncontrolled inflammation. This is what Dr. Tracy has hypothesized is part of the hyperinflammatory response and perhaps the pathogenesis. I want to suggest something a little bit different. The orexinergic nervous system is something that isn't discussed terribly frequently within the critical care literature, but perhaps it should be. It was brought to my attention by a colleague of mine, a friend, Max Kells, who was looking at the orexinergic system as a potential effector of anesthetics. Absence of the orexinergic nervous system is what happens in patients with narcolepsy. They don't make orexin and they fall asleep at the drop of a hat. The orexinergic nervous system is responsible for stimulating activity, awareness, awakeness. This is a mouse brain. This is the orexinergic system, the red circle. It's a series of about 6,000 neurons living in the hypothalamus, and as you can see, it's got connections all over the central nervous system. All of those have effects on arousal and sleep, and they're very important and have been studied intensely in that particular regard. But they also have effects on centers that are involved with sympathetic outflow and hemodynamics, respiration, pituitary function, and temperature control. All things that become dysfunctional and discoordinated in patients with MODS and chronic critical illness. So one could ask the question, is sepsis a syndrome of disordered orexinergic activity? Well, we decided to look at it in a mouse model. And again, these are animal data, not extrapolatable necessarily to human beings. What we did here was we stained the appropriate part of a mouse brain for orexin, again, isolating the area where these particular sets of neurons live. So the green staining indicates something that's orexin positive. The yellow arrow, red staining, indicates something that's active. When CFOS is expressed in a neuron, the neuron is actively firing. So you can see this particular arrow is pointing to a red nucleus that doesn't happen to have any green around it. So it's an actively firing neuron, but it's not an orexinergic neuron. On the other hand, here's an orexinergic neuron that isn't actively firing, it's just green, and perhaps most importantly of all, here's an orexinergic neuron that's active. Green around, red in the center, expression of CFOS. When we took a series of animals and subjected them to sham operation and harvested brain tissue 48 hours later, at the point in time with this particular model where the animals would be in what, for lack of a better term, we call organ dysfunction or perhaps even chronic critical illness, what you can see in the sham-operated animals, non-septic animals, is a great number of active orexinergic neurons, staining with both red and green. On the other hand, when we use a well-accepted, well-validated model of sepsis, cecal ligation and puncture, you can see that there are a tremendous number of green staining nuclei, but very few that are staining red. So in effect, The orexinergic nervous system is being turned off in this septic model. And in fact, when we quantify these data, we see that under normal circumstances, about 50 to 60% of orexinergic neurons are active. They express both CFOS and orexin. But when we look at the cecal ligation and puncture animals in the columns on the far right, you can see that there's very, very little activity, both at 24 and 48 hours. So, sepsis clearly involves disordered orexinergic activity, at least in this animal model, and MODS could be a disease of the central nervous system. So where does that leave us? Well, it leaves us here. We've got this paradigm, we've got this picture. Certainly we need to add another little component in here, which is to say that at some point during the phase of chronic critical illness, the neurological system, the humoral system, the endocrine system become exhausted, cease to work properly, and perhaps that's what leads to either resolution or death. Certainly, we haven't done all that well in treating sepsis. The hypothesis underlying most of our research in the last 20 years may not be correct. I would suggest that certainly it's only part of the story, and it certainly hasn't led to any big therapeutic breakthroughs. It may be time for a little out-of-the-box thinking, which is to say, let's at least consider that sepsis in MODS is more than an inflammatory disorder and perhaps an endocrine and neurological disorder. On that note, thank you very much. I'd be happy to answer questions.